Good evening, everyone. So this week we will start, but a bit of a solemn note before we start our this week's lecture. We on 9th of October, our country lost one of the arguably the last industrialist who cared about his employees. So I thought that we'll just talk about just a minute, on a person who has touched each of our lives, and the state or the nation was in collective mourning on his passing. So just a few seconds on him that maybe I don't think so that the nation would get any other eminent personality to replace the loss that it has underwent. And I just thought that we'll start this week's presentation as a tribute to him. Not a tribute to him particularly. We are not doing anything that would tribute a person of that stature. But just to remember him by that people like him are seldom and far between. They do come and go when the work is done. So we'll just remember all the good works that he had done for the nation. Okay. So let's start this week's lecture, and this week we'll discuss keratinization. Let's start. So, as I've asked a lot of people now that what topic should we gather next, and a lot of the votes come for keratinization, and it's a very, in my opinion, it's a very easy concept to grasp. Okay, keratinization is not that difficult. All you need to remember is what exactly is happening as the cell moves from the basal layer to the stratum corneum. Okay, so we are going to deal up with epidermal cells, and as a new the cell which is formed at the basal layer at stratum basale, as it moves slowly towards stratum corneum, what changes happens inside the cell that makes it lose its organelles, that forces it to become dead and harden and become full of keratin, that it forms the stratum corneum layer, is what exactly is keratinization. Okay, keratinization is a process through which a cell from the basal layer gets modified, gets transformed as it moves to stratum corneum and becomes hard. Okay, that essentially is keratinization. What happens to the cell it at each and every step as it moves from the basal layer to the stratum corneum is what we are going to study now. Okay, so without much further ado, let's start this week's discussion on the basics of keratinization. Let's start. Now, what is keratinization? Keratinization is nothing but the formation of the cornified envelope of keratin. Okay. So, as a cell moves from the basal layer to the stratum corneum, it undergoes a lot of changes, and the collective changes, are known as keratinization. Okay. Is known as the process of keratinization. So we have a movement of the cell from stratum basale where the keratinocytes proliferate. That means they divide. Okay. The stratum basal is the generating layer. It generates new cells. Okay. As the cell moves to the granulosome layer, you have essentially all the materials ready for keratinization. Okay. The cell has collected or formed all the proteins required for it to become a corneocyte. Now, corneocytes are the cells that we call in the corneocytes. Okay, corneocytes are the cells that we call when it reaches the stratum corneum, when it has formed the cornified envelope. Okay, so that is the formation of a corneocyte from a keratinocyte is the process of keratinization. And as the cell moves from the basal layer through the granular layer towards the stratum corneum, you have the formation of the cellular envelope or the cornified envelope. And that is the process that we'll study. Let's start. So, in order to understand keratinization, it's easy to understand if you look at the formation of cornified envelope like we build a wall. Okay, so in a wall you have bricks. In a wall you have cement. Okay, and when you put cement or you put concrete, let me just tell you. Imagine the wall above your head as the cellular membrane. Okay, as the plasma membrane. Okay, inside the plasma membrane, there are a lot of iron rods. Okay, these rods are there in your roof. Those iron rods are keratin intermediate filaments. Okay. Surrounding the iron rods is RCC or reinforced cement, concrete cement, and that is made up of concrete and gravel. Okay, gravel or small stones. So the small stones are the collection of epidermal proteins, while the concrete is made up of proteins like loricrin, filaggrin, involucrin. Okay, so this mixture of all the epidermal proteins reinforcing the rod-like keratin intermediate filaments forms the cornified envelope. Clear? We'll understand each of them in detail and how they are formed, and then your understanding of keratinization would be easier and it would be cleaner and it would be better. Okay, so let's start first with the formation of keratin intermediate filaments. Okay, so let's start. So in human cytoskeleton, that means the skeleton of the cell of humans, it is composed of majorly microfilaments from 5 to 7 nanometers, microtubules from 20 nanometers, and intermediate filaments which come in between these two. That is why they are known as intermediate filaments with a diameter of about 10 nanometers, roughly 10 nanometers. So this is the diameter that we are talking about. Okay, in the one in the middle, the green one is the intermediate filaments. Now those are about 10 nanometers, and we are going to discuss, we are going to limit our discussion to intermediate filaments. Okay. So in this cytoskeleton filaments, we're talking about intermediate filaments. These are filaments which are about 10 nanometer wide, and they consist of a lot of different proteins, one of them being keratin. Okay. Other proteins like vimentin, lamin is also not to be confused with laminin. Lamin is different. Now, the function of intermediate filament is to maintain cell shape by resisting tension or pull. So, what exactly we're talking about is intermediate filaments are cytoskeletal proteins or cytoskeletal filaments responsible for maintaining the shape of the cell. Okay, so these filaments they go and attach themselves from the nuclear membrane to the cell membrane, from the cell membrane to the cell membrane, to and these maintain the shape. So whenever you see in a diagram or in a biopsy or in a histopathology, circular cells, they remain circular because they have attachment via the intermediate filaments. That is how the cell keeps its shape. Going back to our example of the roof and walls, your room maintains its shape because it is held together by the cement, concrete, and the iron bars. In the same scenario, the keratin intermediate filaments and the proteins maintain the shape of the cell. Okay, so the function is to maintain the cell shape and to anchor nucleus and some other organelles. That means to keep the organelles in their place so that they are not, freely mobile and proper transfer of materials between the organelles or among the organelles can take place. So that is the function of intermediate filaments. Now these intermediate filaments are roughly of 6 types and they are encoded by 70 genes. And 54 out of these 70 genes encode keratin intermediate filaments. Remember, I told you that intermediate filaments are of different types, lamin, vimentin, nestin. These are all different types of intermediate filaments. So there are total 6 types of intermediate filaments, and all of them are encoded by 70 genes, a total of 70 genes, and 54 out of those 70 genes encode for keratin intermediate filaments. So this could be a good quiz question, okay? Let's move forward. And look at this intermediate filament structure. These are proteins, okay? And what are proteins? Proteins are long chains of amino acids, okay? We know that. Now, amino acids, if they are strung together, if they're collected together, one pole will have the carbon terminal, the other pole will have the amino terminal, okay? So you have a protein which starts from the carbon terminal and goes to the amino or the NH3 terminal, amino terminal. And this essentially is what a protein molecule is. Okay. If you entwine, if you collect two protein molecules and helically wrap them around each other, you form a dimer. Okay. So a dimer has two keratin molecules. two protein molecules. Okay. Now, if you attach two dimers together, you form tetramer. Okay. And if all the carbon molecules, all the carbon terminals are together, then it is a parallel. And if carbon and amino terminal alternate with each other, so you can here see that in the tetramer of intermediate filaments, the carbon and nitrogen or the carbon or amino terminals are alternating with each other, and that is known as a staggered configuration. Okay. Now this staggered configuration forms a tetramer. Okay. Now these tetramers two, three, four, five. This tetramers, you can see this is a tetramer, and this collection of tetramers forms protofilaments, keratin protofilaments. And eight of these protofilaments, okay, eight of these protofilaments come together to form the keratin intermediate filament. So now you understand. Protein molecules, two protein molecules form a dimer, two dimers form a staggered tetramer, staggered tetramer forms a protofilament, eight protofilaments form intermediate filaments. Clear? So let's move forward. Now these are the six types of intermediate filaments that I was talking about. Type 1 and type 2 are the keratin in the skin. Okay, so we are going to restrict our discussion to type 1 and type 2 keratins. Type 1 are the acidic keratins, while the type 2 are the basic keratin. Type 3 include desmin, vimentins, peripherin. These are all mesenchymal origin proteins. Type 4 are neurofilaments and related proteins like alpha-interleukin. Nestin protein in the eye is a type 4 intermediate filament. Lamins are the nuclear intermediate filaments. Okay. And fecinin and filancin are eye proteins, which are the type 6 intermediate filaments. But we are going to restrict our discussion to type 1 and type 2, which are the keratin intermediate filaments. Okay. Let's move forward. Now, how do we classify keratins? If you want to study something, you need to classify, group them together. So Moll et al. In 1982 classified them into type 1 and type 2, and that was on the basis of their chemical structure. Okay, so this was acidic, and this was basic group of keratins. Clear? In 2006, this classification was revised, and this was revised by Rogers et al. And keratins then were classified on the basis of their chemical properties. So you had epithelial keratins, which deal with skin, and you have hair keratins, and in fact nail keratins, which deals with the hard keratinization. Okay, so keratinization, the process of keratinization is essentially of two types: soft keratinization, which happens in skin and mucosa, and hard keratinization that happens in hair and nail. Okay, let's move forward. So we're going to discuss type 1 and type 2 keratin, and these are the differences between type 1 and type 2 keratin. Type 1, as I've said, are acidic, while type 2 are neutral or basic. Type 1 is found in chromosome 17. Type 2 is found in chromosome 12, the genes to encode keratins. Type 1 has lower molecular weight, about 50 to 64 kilodaltons, while type 2 has higher molecular weight, about 52 to 67 kilodaltons. Okay. Homologous domains. We'll discuss what are homologous domains in few slides afterwards. They are absent in type 1 keratin but present in type 2 keratin. Okay. So we have till now understood that there are two types of keratin, 1 and 2. One is acidic, 2 is neutral basic, and these two are different. What happens is that during the formation of the dimer, type 1 and type 2 exist together. For example, one strand of the dimer will be made by type 1 keratin, while the other strand of the dimer will be made by type 2 keratin. So in a dimer, one part will be made by type 1 and the other part would be made by type 2, and this dimer exists in the tissue as keratin intermediate filaments. Clear? Now, what are the tissue expression patterns? As I've said just now, that keratin dimer forms from one unit each of the both types of keratin. So you will have type 1 keratin and then you will have type 2 keratin, and both of them together will form a dimer. Okay. The pairing depends upon the molecular weight of individual keratin. That means these dimers are specific. For example, keratin 1 can attach to keratin 14. Keratin 1 can attach to keratin 2. That is not an issue. Keratin 1 can attach to keratin 38. But what kind of dimer you will see depends on what tissue you are observing. For example, different dimers would be present at the basal layer of the skin. Different dimers would be present at the outermost or the uppermost layer of the skin. Different dimers in mucosa. Different dimers in nails. Different dimers in hair. But remember, each dimer will have one keratin from type 1 and one keratin from type 2. Okay. And that is known as expression to be tissue specific. That means if you're looking at a tissue, if you're looking at hair, you will have different dimers. If you're looking at nail, you will have different dimers. Okay. So every dimer is made by the attachment of one type 1 keratin with another type 2 keratin, and they both exist as dimers. Clear? So we have reached from keratin genes to keratin dimers. Okay. Let's move forward. Now, to understand the process of keratinization, we will discuss the keratin intermediate filaments and its formation. Then we'll discuss the proteins of the epidermal differentiation complex. Then we'll discuss transglutaminases, which are very important enzymes for the formation of the cornified envelope. We'll see keratohyalin granules and lamellar bodies. And then we'll discuss calcium and proteases, which are required for the process. So one by one, we're going to discuss these parts. Let's start. So let's start with keratin intermediate filaments. So prior to this slide, we have made the dimer. We have reached the formation of dimer. And we know that these two dimers will come together in an opposite fashion in an anti-parallel tetramer or a staggered configuration to form a protofilament. And 8 of these protofilaments will come together to form keratin intermediate filaments. Okay. This dimer is also known as coiled coil dimer or a coiled coil because they are two coiled together. So this is another name for keratin intermediate dimer, not filament, dimer. Okay, let's move forward. So we were discussing the structure of keratin. This is a protein molecule or the keratin molecule, and we have the amino end here and a long chain of amino acids, and then you have the carboxy end. Clear? That is how a normal protein structure looks like. Basic structure. Okay. Now, amino end, carboxy end. You have a tail portion near the carboxy end and you have a head portion near the amino end. In between, you have a thicker portion which is known as rod domain. Rod domain has four parts, 1A, 1B, 2A, 2B. And these parts are separated by short strands of amino acids, and these are known as linkers. Okay, so linker. Now what happens is that you have the amino end, you have the carboxy end, you have the chain of amino acid. It becomes helical, 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 and helical. Okay. Now these are the rod domains. Rod domains. And wherever the helix start, let me change the color of the pen. So wherever the helix start is known as helix initiation motif. Okay. And wherever the helix ends, let me change the color. Wherever the helix ends, here it is ending. So it is helix termination motif. Clear? We're clear about it. So you have a single chain, then helix formation is there, and wherever the helix start, it is a helix initiation motif. Where it ends is the helix termination motif, and in between you have the linker DNA. And these all areas are the mutation hotspots. That means any changes in the amino acids here or there or in linker DNA or in termination motif will change the amino acid reading pattern of the chain and will lead to different protein formation. That is why you have the mutation hotspots in this protein. That is why you have different keratin mutations leading to different kind of disorders. Okay. So if we simplify the structure, we can say that you have the head portion, the tail portion, and a thick alpha helical rod domain in between. Clear? So this is the simplified version of the keratin gene or a keratin molecule. And these alpha helical rod domain has heptad repeats. Now what are heptad repeats? It's A, B, C, D, E, F, G. Each of these letter is a different amino acid. Okay. And since there are 7 amino acids, it is heptad. And it keeps on repeating. It keeps on repeating. That means A, B, C, D, E, F, G, A, B, C, D, E, F, G, A, B, C, D, E, F, G. It keeps on repeating. And these are known as heptad repeats. So all of these heptad repeats are found in the alpha helical rod domain. Okay. So now we are back to the structure of keratin. We have the head portion here, and we have the tail portion here, and we have the alpha rod portion here. Okay. So this is the alpha helical rod domain, and these in between are the linker DNA. We have discussed that. These are the helix boundary motif. If it's at the starting, then it is the initiation motif. If it's at the end, it is termination motif. Okay. Now, in alpha helical rod domain, it is a highly conserved portion. Conserved means that the mutations are very well taken care of. Okay. And it is a very important a part of the protein molecule. If there are any changes in the amino acids there, the keratin will not be able to function properly. Okay, so it's a highly conserved. That means in nearly all the properly functioning cells, you will find that they are same. There's not many changes occur in the alpha helical rod domain because it's a very important part. So we have heptad repeats, 7 amino acid residue repeats, keeps on repeating n number of times. Okay. Yes, and A and D, that means the first amino acid and the fourth amino acid are hydrophobic residue because they are used for stabilizing the heterodimer. Heterodimer are, remember when the keratin dimer forms and then the dimer gets connected to another dimer forming a tetramer and they're in a staggered position. To stabilize the tetramer structure, you need to have some hydrophobic residues, and that is what the function of A and D is. So they are responsible for assembly of intermediate filaments and for structures. Okay, let's move forward. Now this H2 part is present type 2 keratin. Remember we discussed when we were discussing the table between type 1 and type 2 that you have the domain present in type 2, not in type 1. This is the domain we are talking about. Now in helix boundary motifs, these are mutation hotspots because if any problem happens in the starting, okay. Now let me explain like this. In a gene, you have the promoter sequence. At the promoter sequence, the cell knows that I have to start reading the gene from that point. Then you have the termination sequence. So the cell knows that I have to stop reading the gene at that point. If any problem happens in the promoter sequence or the termination sequence, the cell will not know when to start and when to stop. And if the cell doesn't know when to start or when to stop, altered genes will be created, altered proteins will be created, the proteins will not be normal, and that will lead to mutation. Similarly, if there are mutations found in the alpha helical rod domain initiating amino acids or alpha rod domain terminating amino acid, thus the protein will not know when to start the helix and when to stop the helix, and this will lead to an altered keratin. Okay, altered keratin formation. And this altered keratin formation will lead to mutations, will lead to diseases. Clear? Now we know that alpha helical rod domain is highly conserved, so most of the mutations or typically the mutations are seen in the helix boundary motifs. So mutations in these motifs result in more severe disease. Just go back and see our video on epidermolysis bullosa junctional, and you will realize that there are some types of junctional EB which are very severe. In most of the time, the mutations are happening in the keratins at the level of helix boundary motifs. So if the mutations are there, if the keratin is mutated because of problems in these boundary motifs, it is much more severe. Okay, let's move forward. Now, remember I told you that the expression of keratin dimers are tissue specific. That means depending on what area you're looking at, you will have different dimers. So that is what we are trying to show with this diagram. Okay. So you have simple epithelia. That means simple epithelium. You have these keratins. I have used different color coding for type 1 and type 2 keratins. So simple epithelia has these keratins. In the basal layer of stratified epithelia, that means layered epithelia, in the basal layer you have these keratins 5, 14 and 15. You remember 5, 14 from our discussion on epidermal bullosa, that these are the keratins at the basal layer of cells. You also have 6b and 17, and that is much more important in nails. These are hard keratins. Let's move forward. Now 6a and 16 are seen at the upper layers. So in a skin, you have the basal layer, granulosum, or in fact first spinosum, granulosum, and corneum. Okay, so in the basal layer you will have 5, 14, while the upper layer will have 1 and 10. In the case of nails, the upper layer have 6a and 16, while the basal layer has 6b and 17. Clear? In the epidermis, you have 1 and 10. You can also have 2 and 9. In mucosa, you have 76 and 12, which is non-cornified mucosa. In the cornified mucosa or thicker mucosa, you have 4 and 13, while in cornea, you have 3 and 12. Okay, so this is the expression of different dimers in different tissues. And mutation in individual dimer will lead to clinical manifestation of diseases. For example, if there is a mutation in keratin 6a or 17, like we see in Pachyonychia congenita, you will see involvement of nails. If there's mutation in 1 or keratin 5, you will see epidermal bullosa. Okay, so depending on what keratin is getting mutated and its localization, you will have clinical effects, clinical features of the disease. Okay, for example, mutation 5 and 14 is epidermal bullosa simplex. Dowling Digos, there's mutation keratin 5. In epidermolytic ichthyosis, there's 1 and 10 keratins are mutated. In epidermolytic palmoplantar keratosis, keratin 9 is mutated. In Pachyonychia congenita type 1, 6a and 16 are mutated, while type 2 has mutation in 6b and 17. Now, the current classification of Pachyonychia congenita has, I think, 5 types. The 1 and 2 is not used. But to remember the mutations, we can use see this. Ichthyosis bullosa of Siemens, keratin 2 is mutated. White sponge nevus in mucosa, so 4 and 13 are mutated. Meesmann dystrophy, 3 and 12 are mutated. You have problems in the cornea because 3 are 12 are in the cornea. DPR, dermopathia reticulosa, something like that. I'm not remembering. Keratin 14 is mutated. So now you can understand that different dimers exist in different tissues. Different dimers exist in different layers of tissues. Okay, so let's move forward. The functions of keratin, and this is a very important Viva question or a theory question. What are the functions of keratin? So you can divide the function of keratins into two parts. You have the mechanical functions, that means structural functions, and then you have regulatory functions, that means physiologically functions. Okay. In mechanical function, they maintain the architecture and the cell division. They ensure mechanical resilience of the cell. That means how much the cell is able to resist any changes in shape or structure. Okay, resilience of cells. In regulatory functions, you have help in cell signaling, cell transport, cell differentiation. Remember, keratins are filaments, and filaments are a way for one organelle to connect to another organelle. Okay. Transport of membrane-bound vesicles. So you can use those ropes to transmit material. Helps in melanosome distribution in keratinocytes, like keratin 5. Maintenance of hair in anagen stage, like keratin 17. Stress control, and stress control is not the general stress, stress to the cell to control that. And cornified layer is a rate limiting membrane for passage of water. So this reduces transepidermal water loss. So in many disorders like atopic dermatitis, when you have high transepidermal water loss, these cornified envelope is the one that actually stops water from losing from your skin. Okay, so it's very important. The formation of keratin and the cornified layer is responsible for maintaining the hydration and moisturization of the skin. Okay, so you have the mechanical function and the regulatory function. Let's move forward. Okay, so we have discussed keratin, we have discussed dimers, tetramers, intermediate filaments. Now we have keratin intermediate filaments ready. Now, keratin intermediate filaments were the iron rods in your walls. Now we'll talk about the formation of cement or the other proteins of epidermal differentiation, and these proteins are collectively known as epidermal differentiation complex. Let's start our discussion. Now, the proteins are localized on chromosome 1. Remember, keratin were on 1 on 17 and 12. Chromosome 1 is where all the proteins of epidermal differentiation complex reside. Okay. Now, these proteins are important component to form the cornified envelope, and they have three clusters. So let's discuss the clusters. Clusters means that genes of multiple proteins occur together, occur close by, so that they are translated together. They are formed together. Okay. So if you look at this structure of chromosome 1, you will have three clusters. You have the trichohyalin and filaggrin, which are the fused gene proteins. You have SPR, involucrin, loricrin genes, which are the precursor of cornified envelope. And then you have the S100 group of clusters, which are the calcium binding proteins. So you have three clustered families, fused gene proteins, precursor proteins, and calcium binding proteins on the chromosome 1. Okay, so this is a good quiz question that epidermal differentiation complex genes are located on which chromosome? They are on chromosome 1. So let's discuss first the fused gene proteins. Now, what exactly are fused gene proteins? Now, each one of this red hexagon will code for filaggrin. It will lead to formation of filaggrin. In fused gene proteins, you have repetition of filaggrin. Okay, repetition of genes that code for filaggrin, and these are very high. For example, here you can see there are hundreds of proteins, and these are just repeated. Why it is repeated? Because when the process of translation starts, when the process of this gene to form a protein starts, you can create hundreds and hundred copies just by translating once. So that is how you can create a large number of proteins naturally. So that because when you start translating the gene to the protein, you go from the first part, you give one molecule, two parts, second molecule, third molecule, fourth molecule. These are all same molecules of filaggrin. But since there are repeats available in the genetic level, you in one single process you form a lot of filaggrin molecules. Okay. So in fused gene proteins, filaggrin is the most important member. You have profilaggrin, which is a precursor protein and gets converted to filaggrin. It's histidine rich and highly phosphorylated protein. Clear? Let's move forward. So profilaggrin, after dephosphorylation proteolysis, converts to filaggrin. Okay, and this filaggrin is an essential molecule for the conversion of keratin to macrofibrils. So what you have exactly is, let me just revise it. You have keratin polypeptides, which are the dimers and the tetramers. They form keratin intermediate filaments. Clear? Now, keratin intermediate filaments, this is the plasma membrane. This is the keratin intermediate filament. Filaggrin will help attach the keratin intermediate filament to the plasma membrane, okay, and form the cornified envelope. So this is the simplest way to understand. So as the cell moves from the granular layer to the cornified layer, you have more expression of filaggrin. Okay, let's move forward. Now let's see the other proteins of cornified envelope. You have involucrin. Okay, you have involucrin, which is rich in glutamates. It's a minor component seen in spinous or the granular layer. It has a role in early formation of cornified envelope. Okay, the loricrin is rich in glycine. It's the most abundant component of cornified envelope, about 65 to 70%. This could be a good quiz question or viva question. The most abundant component of cornified envelope is loricrin. Then you have the SPRP. SPRP are simple proline rich proteins. Okay. So they have an antioxidant properties, and they, along with loricrin, form complexes to attach keratin intermediate filaments. We'll see them in subsequent slides. Don't worry. Other proteins include the statin, keratolinin, elefin, cornifin. The The process of keratinization is filled with a lot of different proteins. Okay, so let's move forward. The third are the calcium binding proteins or the S100 group of proteins. There are total 17 genes and 6 pseudogenes. They are expressed by keratinocytes and the expression pattern changes in various dermatoses. Also, the expression pattern changes as the cell moves from basal layer to the outermost layer, and this leads to formation of a thing known as calcium gradient. And we'll discuss about calcium gradient subsequently, don't worry. The third is transglutaminases. Remember, first we have keratin intermediate filaments. Second, we have epidermal differentiation complex. Third, we have transglutaminases. Now, what are transglutaminases? These are calcium-dependent enzymes and they cross-link proteins with the help of epsilon gamma glutamyl lysine isopeptide bond formation. You may not need to remember this whole. Just remember that transglutaminases are responsible for formation of isopeptide bonds between proteins. So the function of transglutaminases is to form isopeptide bond between proteins. And these are calcium dependent enzymes. These two things you just have to remember. So here we can see that you have protein 2, protein 1, and the transglutaminases attaches glutamine residue to lysine residue. So TG is transglutaminase. Okay. So this reaction requires calcium. So now you know how calcium is important for keratinization and it forms an isopeptide bond. So the function of transglutaminases is to form isopeptide bond between two different proteins by utilizing calcium. Clear? Let's move forward. Now, there are different types of transglutaminases, but we will restrict our discussion to one in the transglutaminase 1, which is found in membrane. It is responsible for cross-linking of cornified envelope precursor. Okay. Transglutaminase 2 is seen in the cross-linking of the extracellular matrix. Transglutaminase 3 is found in the cytosol. It completes the cross-linking of cornified envelope. Exactly how it acts, we'll tell you, don't worry. And transglutaminase 5, the important point to remember about TG5 is that mutation leads to acral peeling syndrome. So this is a Viva question or a MCQ question. What mutation is seen in acral peeling syndrome is transglutaminase 5. Let's move forward. The fourth part after transglutaminases are the lamellar bodies. Now the function of lamellar bodies is to transport pro-barrier lipids to the outermost layer. Now, till now, we have studied that the formation of a cornified cell from a basal cell. That is what we have studied. But in between those stacks of cells, you have lipids. And what is the source of lipids? The source is lamellar bodies. These are responsible for carrying the lipids. Okay. It also carries other enzymes like cathepsin D, acid phosphatase, glycosidase, protease inhibitors. Okay. So, lamellar bodies, let me just make a big lamellar body has majorly lipids with few enzymes. And what acts as the signal to secrete these lipids inside in the cornified layer is the calcium gradient. Remember we talked about calcium gradient, okay? And because of the presence of this calcium gradient, these lamellar bodies know that they have to extrude out, okay? And look at this electron microscopic picture. You will see this area, these lamellar bodies or odd-land bodies. Okay, these lamellar bodies are filled with lipids ready to be secreted outside. Okay, so let's talk about the calcium gradient. Now, a gradient exists between the basal layer and the stratum corneum. So, as the cell moves from stratum basal to stratum corneum, you will see that the calcium concentration slowly increases. At the level of stratum granulosum, there's a spike. And this spike acts as a trigger for lamellar bodies to get secreted. If you remember your physiology, you will understand that calcium influx is attached to the efflux, the outside flux of vesicles, isn't it? Whenever there's calcium influx, the vesicles are discharged. Similarly, the lamellar bodies get their signal to discharge because of a calcium gradient. Okay, we're clear about this? Now, this calcium gradient is responsible for differentiation of keratinocytes because look at this. A lot of things are happening at the level of stratum granulosum. And we know that there's a spike happening. So there must be a relation that from this point onwards, the cell decides that now I will form a corneocyte. I will not go back. Okay. Other molecules like protein kinase C and the calcium-dependent enzymes transglutaminases are activated in the presence of these calcium spikes. So this calcium gradient is very important. This could be a very good short note that what is the role of calcium in the process of keratinization. Let's move forward. Now let's look at the role of calcium. As the calcium gradient increases from the basal layer to the corneum layer, the basal cells are also increasing in size and metabolic activity. Why metabolic activity? Because they are trying to make a lot of proteins now required for cornification. The intermediate filaments are increasing and aggregating in the inner lamina of plasma membrane. So this is the plasma membrane. The outside portion is the outer lamina and the inside portion is the inner lamina. Clear? Now, calcium is sequestered in proteins. And what are the proteins? The major one is the S100 group of proteins. In the endoplasmic reticulum. Okay. Now, what exactly is happening as the basal cell is moving towards the stratum corneum? It is forming a lot of proteins which are required. It is also collecting calcium. So, if you remember our graph from the last slide, the calcium is slowly increasing. And this is because it is collecting slowly calcium in ready to be discharged the moment it reaches calcium, stratum granulosum. We can see that in stem cells, the organelles are starting to get destroyed. Why destroyed? Because the cell is now slowly removing its organelles in order to form a corneocyte. Clear? The cell eventually forms TAC or transient amplifying cell. Okay. Constantly, as the cell is moving from basal layer to the upper layer, the calcium gradient increases, the calcium influx, the calcium concentration in the cell increases, and the calcium guided enzymes like transglutaminases and proteases are also activating. And because of the activity of proteases, the cell structure starts getting degraded. Okay, proteases will destroy proteins, so cellular proteins are being destroyed. So you can see that here the blue one is actually cornified envelope and it has started to form. We can see that the organelles are getting destroyed and we can see that the calcium ion is increasing inside the cell as the cell has formed transient amplifying cell. Clear? Let's move forward. Now, when the cell reaches stratum granulosum, and the sudden influx of calcium has happened, and this is the calcium spike that I was talking about. The cell has decided to become a corneocyte, and now it is known as a committed corneocyte. And you can see that the cornified envelope is formed. It's thicker, it's formed. You can see the cell is filled with calcium ions because an influx has just happened from the endoplasmic reticulum. You can see that the nucleus is somewhat destroying. It's getting pycnotic. It's getting condensed and destruction. Organelles have disappeared and the cornified envelope is complete. So remember, basal cell starts collecting calcium inside smooth endoplasmic reticulum, binds to S100 protein. As the cell moves to upper layers and reaches stratum granulosum, all the calcium ions are released simultaneously. This release of calcium ion uh, triggers the formation of a corneal envelope and the cell becomes a committed corneocytes. Okay, let's move forward. Now in corneocyte, the organelles have disappeared, the envelope is fully formed, the extracellular lipid envelope also formed, and this lipid has come from lamellar bodies. And why this lipid is there? Because calcium influx has happened leading to discharge of lipids. The cell eventually dies because there are no organelles. It is just a cornified envelope. And now it has become a corneocyte. So from a committed corneocyte to a proper corneocyte. So this is a corneocyte, a dead cell with the cornified envelope inside and a lipid envelope outside. So you have the outside lipid layer formed by the lamellar bodies and you have the cornified envelope inside formed by proteins. Let's move forward. Now, proteases are very important. We talked about transglutaminases. We have talked about calcium ions. We'll put just one or two slides on proteases. Now, proteases are required to cut the protein and activate them. Remember, like filaggrin gets converted to filaggrin. So this all conversion happens because of proteases. They're responsible for differentiation of proteins. They can degrade unwanted components, and they're responsible for organelles also getting destroyed as the cell is moving upwards, and they are required for regulation of component assembly. That means they need to take care of all the different proteins that have been formed but are not required. So that is a function of proteases, and we know that a lot many proteases are calcium dependent. They get activated because of presence of calcium. One of the important proteins are protein kinase C, which is activated in the presence of calcium gradient. It downregulates keratin 1 and keratin 10. Now remember, keratin 1 and keratin 10 are the important keratin dimers seen in all layers of epidermis except the basal layer. In the basal layer, you have 5 and 14, while in the upper layers, you have keratin 1 and 10. Protein kinase C also activates markers of the granular layer. That means the uh, the granular layer markers is there. When the cell reaches the stratum granulosum, you have why the layer is known as granulosum, because the cell has a lot of granules, and formation of that granules requires the activity of protein kinase C. And remember, the calcium gradient spike is also occurring at the level of at the level of stratum granulosum. Okay, so at the level of when the cell reaches stratum granulosum, in the presence of calcium gradient spike, protein kinase C gets activated, leading to all the different functions of proteins. Okay. Now these functions include protein induction like loricrin, filaggrin, etc. So all the proteins of cornified envelope starts forming. So with that basic understanding, we know the players, we know what is happening. Let's discuss keratinization and the process of keratinization. Okay. Now look at this diagram. This I've taken from Rooks 10th edition, the latest edition on page 2.7. And this is a very good diagram, but in my opinion, it's a slightly bit complicated. Okay. So we'll start from here. We have the desmosome. We know desmosomes are the connecting molecules or the connecting group of proteins which connects two cells together. And in this you have keratin filaments. So these are keratin filaments. We know that the presence of calcium is required for the attachment of protein molecules to the cornified envelope. So this here the attachment is happening. Okay. The attachment is mediated by enzymes like transglutaminases. When the attachment is happening, you have the secretion of lipids by the lamellar bodies. And this lamellar bodies will form a lipid envelope around it. Clear? And as we move forward, we have the strengthening of keratin intermediate filaments by the addition of SPRs and loricrin. Okay? And this loricrin granules are found in keratohyalin granules. Okay, and this leads to formation of, if we first see the inside of plasma membrane, you have loricrin, involucrin, you have filaggrin, and keratin intermediate filaments. If you go outside the plasma membrane, you have the lipid envelope, and this together forms the cornified layer. Okay, so this is a very good diagram aesthetically, but I think it's a bit complicated. So let's simplify the process so that you're able to understand. Clear? You have the plasma membrane. You have the desmosome. Desmosomes are connecting molecules through which the cells attach themselves to other cells. Okay, desmosomes. You have intermediate filaments, which are the keratin intermediate filaments attached to desmosomes. Clear? Then comes periplakin and envoplakin. These are other attachment molecules. Then you have involucrin, which is the protein of cornified envelope. Under the action of transglutaminase 1, involucrin attaches itself to envoplakin and periplakin. Same thing is happening at other areas also. So under the action of transglutaminase 1, other areas are also getting attachment of periplakin, envoplakin, involucrin. Clear? Now, second step. There will be formation of cornified envelope. That means inside the plasma membrane, in the inner side, you have cornified envelope. You have the keratohyalin granules now. The keratohyalin granules gets degranulated. and the filaggrin and loricin proteins are now out. Okay, and this is the loricin granule that we saw before. Loricin granule. Clear? Loricin granule, along with SPR, small protein-rich peptides, SPR forms complexes, this dumbbell-shaped complexes. A lot of these complexes, by the action of transglutaminase 3, gets attached at the underside of plasma membrane, the inner side of plasma membrane. Clear? Along with that, the filaggrin also gets attached to the inner side of plasma membrane. And if you remember the starting of the lecture, I have said that the function of filaggrin is to attach keratin filaments to plasma membrane. Okay, so let's attach keratin filaments to plasma membrane. So this is formation of cornified envelope inner side. Clear? And the role majorly is played by the calcium ion influx and the activation of proteases. Let's move forward. So now you will have the formation of lipid envelope outside it. You have the lamellar body, and this lamellar body, along with the action of calcium gradient will release its ceramide lipids, and this lipid will form a lipid envelope outside the plasma membrane. And with the lipid envelope outside and a cornified envelope inside, this is the formation of your cornified envelope. I hope I've made the process clear to understand, simpler. So a keratinocyte with degrading granules, you have the degenerating granules, degenerating organelles, you have the extracellular lipid envelope formed by lamellar bodies, you have the cell membrane in between, you have the intracellular cytokeratin envelope. Slowly, with time, this will become flatter to form corneocyte. The organelles have gone, the cell membrane has gone, you have extracellular lipid envelope and a corneocyte or a corneal envelope, cytokeratin envelope. So that is now a corneocyte. Clear? Let's move forward. Now, we have finished keratinization as of now. We'll discuss very shortly on defects in keratinization. Of course, each of these require their individual discussion, so we'll be very short and discuss this. So they can be classified into two types. Either there is mutation in the keratin genes or there aberration in the process of keratinization. So either the keratin genes are not working properly or the process is not working properly. So you can just imagine that either the cement is not good, the bricks are not good, or here the mason is not good. Okay, so let's move forward. So this is a diagram that I've taken from Bologna 5th edition, page 887, and it just tells you that from where different locuses are there for mutation for different disorders. For example, if the mutation, I'll just tell you how to read this chart. In the helix initiation motif, if the mutation happens in the HIM of keratin 4 and 13, you have white sponge nevus. Okay. If the mutation happens in the HIM of keratin 9, you have epidermolytic palmoplantar keratosis. Okay. If the mutation happens in keratin 17, you have pachyonychia congenita. So now you understand how to read it. The area remains the same, but depending on which keratin's area is mutated, you have different disorder. For example, in pachyonychia congenita, you have mutation 6B also in different type of pachyonychia congenita, and here it is the mutation at the termination motif. Okay, mutation in V2 causes ichthyosis hystrix of Clerth and Maclean. Okay, so all of these areas, mutation in the linker DNA is causing localized EBS. If you remember, we have discussed that in epidermal bullosa. So just to understand how to read this diagram is that in different areas of different keratins and mutations happen, what all disorders you will see. Okay, let's move forward. I've tried to simplify the table so that you should remember few of the important examples. Okay, so if the component of epidermal differentiation complex is mutated, for example, if the defect in loricrin, you have loricrin keratoderma or progressive erythrokeratoderma. If the keratohyaline granules are mutated, for example, the filaggrin is mutated, you have ichthyosis vulgaris. If lipid transporter like ABCA12 is mutated, you have Harlequin ichthyosis. If lipid synthesis is wrong, then you have X-linked recessive ichthyosis, Sjogren-Larsen, or child syndrome. And here the mutation is happening at the level of lamellar bodies. If transglutaminase 1 is defective, you have lamellar ichthyosis. If transglutaminase 5 is mutated, we have axial peeling syndrome. If ATP2A2 is mutated, you have Darius. If ATP2C1 is mutated, you have Hilly-Hilly disease. If SPINK5 proteases is inhibited or mutated, you have Netherton syndrome. So I've tried to take out, the disorders that we usually hear about in our OPDs and discussion and try to form a table so that it's easier to understand and remember. So further reading, these are the chapters that I've used along with a lot of different sources, a lot of different articles, and you can go through them if you want. But if you have gone through this properly, this video lecture, I hope the process of keratinization is clearer. Now, one important announcement that I have is that NAMSCON MediQuest 2024 is going to be there this month. And it's a national level quiz organized by the National Academy of Medical Sciences. The prelims are happening on 27th October and the prelims are entirely online. I'll put the link to register. The registration is free but mandatory. You have to be registered. Otherwise, you will not get the link to start the quiz. And it's free of cost. The link is in the description of this video and you can scan this QR code to get directly to the Google form to register for the quiz. On 27th October, we'll be having an online quiz at the countrywide level, and the finalists will be chosen to take part in the finals NAMSCON quiz. And if you have heard of this quiz before, they have exciting prizes, and I am one of the quiz masters which will be contributing quizzes and organizing the quiz at the national level. So make sure you scan this QR code, get registered yourselves. The registration is free, but you have to register. Give the quiz in an online mode on 27th October, and maybe you'll get selected to finals if your scores is good enough, and we'll meet you in finals personally. Okay, so remember, scan the QR code, get registered yourself, and I hope that a lot of people who have gone through this video lecture and reached the end scan for it and go for quiz. The questions are from medicine and a major chunk of the quiz is general knowledge quizzes or non-specific, non-medical questions. So people who maybe in your family, if your siblings or other people are not from medicine background, they can easily get registered because it goes from different domains like medical science, current affairs, mental health, movies, sports, and history. So a lot of different domains will be tackled. Scan and register, and I shall see you in the finals of this quiz. We'll see you in prelims also. The prelims is on 27th October in an online fashion. Let's meet you there. So with that, we will end our discussion on keratinization. It's a process which is easy to understand if you know what all players are working and how the envelope is forming. It's a very interesting process. In fact, I find keratinization to be a lot more interesting than melanogenesis. Okay. In fact, my thesis was in vitiligo, but still I found keratinization a much more interesting process to read about and understand. I hope I have made the process a bit clearer. You can comment all your doubt suggestions with timestamps, or you can email me, but it's always better to comment. And with that, adios, and thank you for watching this long lecture. Bye bye.